Hello, welcome to this video where we'll be dissecting a sheep brain in order to learn more about the structures that we have been covering this week. But before we start, it is going to be important that we discuss a little about the ethical aspects involved. And you are more than within your rights to have any stand that you may have towards using animals in teaching anatomy and physiology. However, what you need to do is that you need to have a justified view and also an appreciation of opposite views. One thing that I want to highlight at this point is that these sheep brains that we will be using came as a byproduct of growing sheep for food production purposes. So these sheep were not grown for this purpose. Their lives were not sacrificed so that we could do dissection. Rather, they would have been butchered anyway and the brain thrown away as a waste. So we are actually using these brains for something useful instead. So that is something to keep in mind when we considering the ethics involved. Let's get started. So here we have a sheep brain with its anterior side facing to the top of the screen and its posterior side facing to the bottom of the screen. We are looking at the brain from the top down. Let's start by identifying what we can see externally. We have cerebrum, which is the large part, and at the back, the cerebellum. We were introduced to this area as the little brain. We can also see the brainstem most posteriorly. So this brain is facing us a dorsal side up. Remember, dorsal was our back, and the sheep's back would be facing this way. So let's make sure that we can identify two cerebral hemispheres, left and right. We can also see jury, the ridges here. And also sulci, the depressions. And the very deep depressions were known as fissures, right? So we can spot the longitudinal fissure separating the right and the left cerebral hemisphere. Okay, well done. Now, I would like us to identify the four lobes of the brain. Remember, the sheep brain has a little different dimensions than the human brain that we have been looking so far, but the same structures are there. So let's start with the frontal lobe. This is where the primary motor area would be located at. Also, it is important for cognitive skills such as planning, executive functions, and if you wish, where I would place our personality. So in the sheep, this is a little smaller than in the humans. Sheep are not known for their amazing personalities or making a lot of long-term plans for the future. Now next, let's look at the parietal lobe. In the sheep, this is large. This is where the primary sensory area is located. So processing the location of the body. For example. So this would be pretty important for the sheep, right? So this is area that is relatively larger in sheep. And next we have occipital lobe at the back. This is where visual information is being processed. So we process the information from the eyes as the first point here and give it meanings. Think of hitting back of your head to the ground. It would be the occipital lobe that takes the most direct impact. And you would be seeing stars. In sheep, this is not hugely large either. And finally, I want to show the temporal lobe on the lateral parts of the brain. This is where the information from the ears is being processed, for example. For example, comprehension of the sounds. In humans, this is where we would have the language processing, both for hearing and speaking. And although this is not as big of a deal in sheep, it is somewhat important, so this lobe is there. Now, we are ready to start gently to look at what else we can see here, other than the most easily accessible external features. Let's separate the two hemispheres at the longitudinal fissure and look down between them. Here we can see a thick band of white fibers. This is our corpus callosum. It is an important structure that passes messages from one cerebral hemisphere to another. And this is how we integrate the hemispheric lateralization, forming more complete experiences. Here's a fun fact for you. In epilepsy, there is an uncontrolled neuronal firing, which spreads across the brain, not remaining localized. And although we now have great medications and other care to keep it better under control, it was not always this good. In the past, one way to treat extreme cases of epilepsy was to cut this connection. And yes, it stopped the spreading of this uncontrolled firing, but it also stopped a lot of other normal information from being passed as it should have. We are lucky to have those days gone. Now next, let's turn the ventral side up. Remember, this is as if we would be looking the brain from the below, from the belly side. Now, let's try to identify the olfactory bulb here. This is an expansion from the brain, serving our sense of smell. And it lies just above the nasal cavity. And we can also see remains of the optic nerves. These would come from here, from the optic chiasm, where the optic nerves cross before the information from the eyes enters the brain. Okay, let's look at the three parts of the brain stem. We can see the midbrain, which is made of the cerebral condyles and our mammary body here. Mammillary bodies serve as the relay station for reflexes to sense of smell. Cerebral epicondyles instead is a group of myelinated nerves. We can also see the pons and medulla oblongata, which then continue posteriorly as the spinal cord. Now we are ready to look at the dorsal structures. Let's look at the brain from the back. And here we can see, if we pull the cerebellum away from the cerebrum, the pineal body hidden inside here. We also see superior colliculi and inferior colliculi. Inferior colliculi is responsible for the movement of head and trunk in response to sound stimuli. Superior colliculi instead oversees the movement of eyes, head, and neck in response to visual stimuli. Pineal gland here, which we also saw here, produces hormone called melatonin at night. Melatonin is the sleep hormone, so this controls the sleep-wake cycle. If you travel over multiple time zones, you might need to take melatonin supplement to adjust your sleeping pattern. Okay, we are now ready to prepare a mid-sagittal section of the brain. So this is going to be a brain that is cut in half. Let's locate here the medulla oblongata, pons, cerebrum, and cerebellum. Now, I want to direct our attention to the cross section of the cerebellum. The inner structure is called the arbor vitae, which translates as the living tree. Finally, I want us to note the corpus callosum. We talked about it earlier as the band of white myelinated fibers that connected the two hemispheres. Thalamus is located somewhere in the center, and we have discussed more about it in our lecture material. Also, note the empty space of the lateral ventricle. And at the back, the fourth ventricle. And finally, the cerebral aqueduct, which lets the cerebrospinal fluid to pass. So these are all the structures that I want us to know for now. Thank you for joining me on this video where we have discussed the sheep brain. I hope you have found it useful to see some of these structures that we have studied in a real brain. All that is left to say is to say you baba. That is goodbye in sheep language.